Thank you for that introduction, Tanya. As she already said, I will be talking today about how we can use transcranial magnetic stimulation, or TMS, to measure inhibitory activity in the brain, both in patients and in healthy controls. I'll start with a very brief introduction to transcranial magnetic stimulation itself. This is a technique which not in its present form, but it emerged in the early 1900s. So since then, there has been an interest in this sort of technique. But in 1985, there was the first stimulator which looks very similar to the stimulators that we use now. We know that neurons themselves communicate with each other using electricity. So there's the resting membrane potential, and when a neuron is stimulated, that changes to the action membrane potential, which travels along the neuron. If we apply an electric field to this neuron, we can, of course, interact with this membrane potential. But the problem with electric field is that if we want to do it through the skin and the skull, it's very uncomfortable. You feel the electric current going through the skin, the subcutaneous tissues, and the skull. And we can use it still, but at lower intensities, not at the higher intensities which we need for certain applications. And this is where TMS comes in, and it uses the principle of electromagnetic induction. So the TMS coil itself has these usually two circles, and there's an electric current moving through those coils of wire. And through the process of electromagnetic induction, this generates a magnetic field. This magnetic field can pass through the skull without causing any pain. And when there is a moving magnetic field, it leads to the generation of a secondary electric field, which can lead to electric currents in any conductor. And in this case, the conductor happens to be the brain itself. So by using the TMS coil externally, non-invasively, we are able to generate an electric field in the brain, which then, of course, interacts with the membrane potential of the neurons. The most common outcome measure that is used with TMS is the motor evoked potential, or the MEP. And the idea here is that we place the TMS coil over the parts of the brain that send outputs to the muscles. Usually this is the primary motor cortex, or the M1. This generates an action potential in the pyramidal neurons, which go to the spinal cord, and synapse on the alpha motor neurons, which then connect to the muscles. Which means is that when we apply the TMS pulse, the muscle contracts briefly, and this is something that we can measure using electromyography placed on the skin. And usually the response looks something like this, and we quantify it as the amplitude from one peak to the next peak. I just said that the MEP is in the end caused by action potentials in the pyramidal neuron, or as it's called here, the corticospinal neuron. But what we know is that TMS most of the time doesn't directly stimulate this pyramidal neuron. Instead, it stimulates a lot of interneurons that synapse onto the pyramidal neurons. These could be intracortical interneurons. They could be coming from the opposite hemisphere, or inputs from the subcortical areas. These neurons tend to have a lower threshold for TMS, so they get activated, and in turn, they stimulate the pyramidal neuron. There are a few studies which were able to record the effect of TMS in the spinal cord itself. And when we do that, what we see is that there is a direct wave, which doesn't always occur. And this is because of the stimulation of the pyramidal neuron directly. But usually we see a number of indirect waves, or I waves. These are because of stimulation of the pyramidal neuron via one of these pathways. In the hand or in any muscle where we are measuring, we don't see these separate waves because they all overlap with each other because of how the muscle contraction occurs, and we just see one contraction. So two takeaway points from this slide. One is that we are usually not directly stimulating the pyramidal neurons, which means that using TMS, we can also learn something about all of these other inputs. And the other thing is that you see a spinal input here. So if there is a change in some sort of spinal interneuron activity, one example would be you are measuring the MEP in the elbow flexor, but at that time the elbow extensor is contracting. Because of spinal circuits, the excitability of the alpha motor neuron can also change. So when we use a single TMS pulse to measure the MEP, what we are measuring is corticospinal excitability. We cannot separate these two based on a single pulse. What I will talk about today is not so much single pulse measures, but usually paired pulse measures, which are often used to look at the activity of gamma-aminobutyric acid, or GABA, which is a primary inhibitory neurotransmitter in the brain. Just a little bit of physiology before I jump into the TMS protocols. As I already said, GABA is an inhibitory neurotransmitter. It is present inside the neurons, intracellularly, cellularly, either in the cytoplasm or in vesicles at the end of the axons. And this is the GABA that is being used as a neurotransmitter to inhibit the postsynaptic neurons. Or it can also be present extracellularly. And this often leads to what is called extrasynaptic inhibition, which is not just communication between two neurons, but a sort of sustained inhibition of neurons in a certain part of the brain. Both of these, even though they are shown in different colors, are showing the same receptor for GABA. But there are actually two different types of receptors. So the receptor on the postsynaptic neuron, which senses the presence of GABA. There are GABA A receptors, which tend to act very fast. And we will see this reflected in the TMS methods that we use to measure GABA A activity. And GABA B, which is slower. So fast would be a few milliseconds, within 10 milliseconds, whereas slow would be something in the range of 100 to 150 milliseconds. In humans, if we want to measure GABA non-invasively, this can be done using either magnetic resonance spectroscopy or TMS. And I just wanted to give a little bit of a comparison between these two techniques and demonstrate why you would choose one or the other in a given situation. MRS is a lot more expensive than TMS. In terms of temporal and spatial resolution, the spatial resolution could be comparable between these two techniques. MRS techniques are also evolving quite fast. But in terms of temporal resolution, TMS offers a much higher possibility of looking at fast changes. And one example would be if we want to look at what happens to GABA activity while someone is moving their finger. We can actually look at what happens just before they move their finger, then when they start moving, and by the time they stop moving their finger. So on the order of a few milliseconds, we can look at the changes in GABA activity. The other thing is that TMS can distinguish between activity at the two different types of receptors, GABA A and GABA B. And since the drugs tend to specifically target one receptor or the other, TMS can be particularly useful for quantifying drug effects. We already know that there is GABA dysfunction in many different neurological and psychiatric conditions, which is also why there are already a number of drugs that interact with GABA receptors. These are usually divided into agonists and antagonists, and this table is just to show you that there is a large variety of GABA active drugs. And some of the other classes of drugs that interact with GABA receptors are, benzodiazepines, barbiturates, anesthetics. So there are already a number of drugs which have been used to look at the effect, look at the effects or the pharmacology of the TMS protocols that I'll be describing next. I'll now start with 4 different TMS protocols that we can use to measure different aspects of GABA. For each of them, I'll describe the protocol, so how we actually measure it, a little bit of the pharmacology, what we know about which receptors it acts on, and then show you a bit of data on what we know about these measures in pathology and sometimes how it even correlates with the symptoms of a certain pathology. The first and the most extensively studied one is short interval intracortical inhibition, or SICI. And I would like you to first focus on the black line here. The TS indicates the test stimulus. This is just it. So this is not EMG data, but a representation of EMG data. And what we would see in the EMG is a sort of artifact just when the TMS pulse is applied. And a while later, we see the motor evoked potential. If this test stimulus is preceded by a conditioning stimulus, in this case it was 2 milliseconds, but it is usually somewhere between 2 to 5 milliseconds, then what we see is that this conditioned MEP is smaller than the test MEP. So in one case, we apply a single pulse. In the second case, we apply 2 pulses, and then we compare these 2 scenarios. What we think is happening is that this conditioning stimulus, which is subthreshold, and by that I mean it is not strong enough to generate a muscle contraction, but it does activate the inhibitory interneurons in the brain. So when the next test stimulus, which is suprathreshold, when this one arrives, those inhibitory interneurons are already inhibiting the pyramidal neuron, and that's why we get a smaller MEP compared to if we only apply the test stimulus on its own. The inhibition is usually quantified as a ratio of these 2 MEPs, the conditioned and the test MEP. There are also a couple of studies which have looked at what happens to the direct and indirect waves in the spinal cord. And what you see here is with the black line, which is a single pulse, you see a number of indirect waves, whereas with the paired pulse, which is shown in green, the later I waves seem to be suppressed. So already in the spinal cord, we can see the effect of these 2 pulses. In terms of pharmacology, there are a number of studies which have looked at SICI pharmacology, and I'm just giving one example here. And in this case, they used benzodiazepines, which are positive modulators of GABA-A receptors in particular, and these tend to increase the inhibition. So in this figure, the upper rows from each of these pairs shows a single TMS pulse, and the lower row shows a paired pulse. The left side being before the participants received a drug, and the right side after they received a dose of this drug. And in both cases, what we see here is before the drug, there is some inhibition, but after the drug, the inhibition is increased. So this was 42% of the size of the test MEP, whereas here it is now even smaller. It's 35% of the size of the test MEP, suggesting that when we increase the activity at GABA-A receptors, we get greater inhibition measured using SICI. There is also some data on nuances about the different subunits of the GABA-A receptor, which I won't go into here, but I've provided the reference in case anyone would like to look into it. In terms of pathology, one condition in which SICI has been looked at is epilepsy. And so sometimes the ratio between the two, the test and the conditioned pulse, are represented in different ways. So in all the graphs, I'll make sure to point out which one is more inhibition and which is less. In this case, 100 indicates no inhibition. What we have on the x-axis is the interstimulus interval. And like I said, the inhibition is seen at the lower interstimulus intervals. At the longer intervals, we see facilitatory activity, which is possibly driven by glutamate and other spinal mechanisms. But if we look right here in this area, the smaller the number, the greater is the inhibition or the inhibitory GABA activity. And what we see here is that patients tend to have a bit less inhibition compared to control. So there's decreased SICI in patients with epilepsy. And there is also some research into how SICI differs between different types of epilepsy. And again, I've provided the reference here. There are changes in SICI which are also observed in schizophrenia, and this one they've helpfully already showed that a lower number indicates more inhibition. And what we see is that compared to healthy controls, especially this last group, which is treatment-resistant schizophrenia, seems to have lower inhibition. There is a trend that also patients who respond to drugs have lower inhibition, but this is particularly noticeable in treatment-resistant schizophrenia. And going one step forward, what this study also showed is that there's actually a correlation between the inhibition and symptoms. So in this case, they looked at symptoms using this particular scale, the positive and negative syndrome scale, in which a higher score on the x-axis indicates greater symptom severity, which seems to be associated with lower inhibition. So less inhibition is associated with more severe symptoms. I'm sorry, this graph is not very clear, but I still like to use it because it shows how many studies have looked at SICI in ALS. And in this situation, again, we see that all the studies consistently show that SICI is reduced in patients with ALS when compared to healthy individuals. There is also an effect of drugs. And in this case, please ignore the labels, but please take my word for it that a greater number indicates more inhibition. This is just represented and measured in a slightly different way. Before we go into the drug, the upper graph shows the difference between patients with ALS who are receiving an anti-glutamatergic drug, riluzole, and healthy controls. And what you can already see is that the healthy controls have a higher inhibition. So there is reduced inhibition in ALS. And when the patients receive the drug for several weeks, initially they seem to have an increase in inhibition. So they move closer to the controls. But by 12 weeks, this effect disappears. The authors here say that it is possibly because of a natural progression of the disease, so that the drug is no longer able to counteract the effects of the natural progression. I'll now switch to a different protocol. This is long interval intracortical inhibition. This is measured in a way quite similar to SICI. In this case, again, we have a test condition where we apply a single suprathreshold pulse, which leads to one motor evoked potential. If that is preceded by a second pulse, but unlike SICI, this first pulse is also suprathreshold, so it also generates a motor evoked potential, and it is now, the two pulses are separated by 100 milliseconds. So with SICI, we see a test MEP, and when there are two pulses, we see a smaller MEP. In the case of LICI, we do see one MEP evoked by this first pulse, but then there is a second MEP which is evoked by this one. And in this interval, we think that there is an activity of happening at GABA B receptors, which leads to a smaller motor evoked potential. So in this case, we are comparing this second conditioned MEP with the test MEP. In this graph, again, lower numbers indicate increasing inhibition, and it's showing an effect of baclofen, which now, instead of affecting the GABA A receptors, is selectively increasing activity of the GABA B receptors. And as expected, if LICI is measuring GABA B activity, we see higher LICI in case after someone receives a GABA B agonist. There are also some interactions between these two processes, SICI and LICI, and this paper talks about that in more detail. In Parkinson's disease, here's a study which looked at both SICI and LICI. The upper graph is SICI. And the first thing we see is that actually it looks like there's no difference in GABA A activity between controls and Parkinson's patients. But if we look at GABA B activity measured using LICI, especially when we use an interval of 150 milliseconds. So I showed 100 in the protocol, but it usually is a range with numbers 100 or higher. And we see here that patients who are both in the on and off state of the medication show lower inhibition than controls. LICI is, well, it's very useful because it looks at a different type of receptor activity, but it has been studied a bit less than SICI, and there's a bit less data looking at LICI changes in patients. So I'll move on to another measurement, which is the cortical silent period. In this case, we are applying a single pulse. So this one is, of the 4 protocols that I discussed, the only one that is not a paired pulse protocol. In this case, the participant or the patient is asked to contract their muscle. In this figure, they're asked to press on a small device. So they have some EMG activity. Once the TMS has been applied, we see a motor evoked potential, and then we see a period of silence. So there's no activity in this period. This is, as you can see, for about 100 milliseconds or so in this example. And participants usually don't notice this, but there is electrical silence in the muscle for a little bit. And this inhibition or the lack of muscle activity is believed to be driven by GABA-B receptors. So unlike SICI and LICI, which are mostly driven by cortical mechanisms. We also know this because we can measure, because of the studies that measure in the spinal cord. So they measure quite high in the spinal cord. In the case of cortical silent period, it is possible that some spinal mechanisms also contribute to the CSP. And the pharmacology is a bit more complex because in addition to GABA active drugs, it can be influenced also by dopamine and serotonin. And it is thought to be mediated by GABA-B, but GABA-B agonists don't seem to have an effect on CSP in healthy individuals. It's possibly because there is some sort of ceiling effect. It cannot be increased any further. However, in patients, we do see some changes in CSP. So this is a case study looking at a patient with dystonia. And this is looking at the effect of a drug baclofen, which, as I said earlier, is a selective GABA-B agonist, which tends to prolong the cortical silent period. So in this patient, we see the dose of baclofen, and we see that at higher baclofen doses, the duration of the silent period increases. In schizophrenia, there are a number of studies which have looked at the cortical silent period in healthy controls compared to patients. But in patients who haven't received any drugs for schizophrenia, there doesn't seem to be an effect. There doesn't seem to be a difference between healthy controls and patients. However, if we look at patients who have received the drug clozapine, which acts via NMDA receptors, but also influences GABA-B activity, then we see that there is a longer cortical silent period in patients with schizophrenia. So we don't really know why the drugs don't have a direct effect in healthy controls, but it seems like the drug effects in patients can be identified and detected using the cortical silent period. The last protocol I will talk about is the short latency afferent inhibition. This is again a paired pulse protocol, which means that we have the same sort of setup. So we have a single TMS pulse that gives us a test MEP. But now instead of a second TMS pulse, there is an electrical stimulus applied to a nerve. So this would be a peripheral nerve in this case, which leads to afferent information or sensory information being sent to the cortex. And this sensory information interacts with the stimulation because of the TMS. So this, as you can see in this case, is the interstimulus interval is somewhere between 20 to 50 milliseconds. We stimulate the nerve, which leads to sensory information traveling to the cortex, and that interacts with the primary motor cortex, changes the excitability of the cortex, leading to a smaller motor evoked potential. Benzodiazepines, which are positive modulators of the GABA-A receptors, seem to surprisingly decrease short interval inhibition, short afferent inhibition. So even though this is a form of inhibition, it seems like increasing the activity of GABA receptors decreases the inhibition. And here we see that, again, lower numbers indicate greater inhibition. So after participants in this case receive benzodiazepines, there is a decrease in the inhibition for a few hours, which returns to baseline later. This might be because SAI is mostly looking at cholinergic activity. So in this case, participants were given a muscarinic receptor blocker, which reduces acetylcholine activity. And in this case, again, we see that there is a decrease in inhibition. The adjusted is just a slightly different way in which they made the measurement. And this seems to be because there is some interaction between acetylcholine and GABA activity, and GABA-A receptor agonists also decrease cholinergic activity. So while most of this is focused on looking at GABA activity, there is the potential to look at activity of other neurotransmitters also using TMS. And SAI has been studied quite extensively in Alzheimer's disease, where we know there is a change in acetylcholine activity. And what we see essentially is that the inhibition, or SAI, is lower in patients with Alzheimer's disease compared to healthy controls. And this has been shown in quite a large number of studies, as you can see here. So anything to the right of this line, which is 0, indicates less inhibition in patients. And as you can see, pretty much all the studies show that there's decreased short afferent inhibition in patients with Alzheimer's. And more importantly, the effects of giving drugs to counteract Alzheimer's disease seems to also have an effect on SAI, which is correlated with any sort of improvement in symptoms. So what they've done here is looked at the change in SAI and counted how many tests the patients improved on. The various tests used to look at the symptoms, and it seems, that patients who showed improvement on multiple tests also showed a bigger change in the short afferent inhibition. So a drug that increases the short afferent inhibition could potentially be correlated with actual symptoms, which is what we are eventually interested in. In summary, TMS offers many possibilities to measure neural inhibitory activity, often driven by GABA, but also by its interactions with other neurotransmitters and mechanisms. Changes in TMS-based measures of GABA activity have been demonstrated already in a wide range of pathological conditions. And there is also evidence for the value of TMS-based GABA measures for detecting drug effects, which might also be correlated with symptom alleviation. That is all I have. Thank you. And I'd be happy to take any questions.